Did anyone have any problems with the website, or is everything good there? I saw you guys all made your passwords and stuff. Also, Tanya, could you turn on your camera? Okay. So today we're going to be going over protein quality control. Oh, I haven't shared my screen. I'm done. There we go. Okay. So today we're going to go over protein quality control, continued, and then the DNA damage response. So the DNA damage response is a very complex multi-proteomic cellular cascade problem that is linked to many problems in the cell. So what can cause and start the DNA damage response? Well, spectrum of stresses. So everything from environmental factors such as irradiation and chemotherapeutics, to things such as replicative stress and transcriptional stress, epigenetic dysregulation, to other parts in the cell such as ER stress, mitochondrial stress, and reactive oxygen species. So, just a review of the unfolded protein response. So the unfolded protein response is like the, I actually want to pull up your guys' worksheet to make sure I'm hitting on the things that are on the worksheet. Okay. So what is reactive oxygen species? So reactive oxygen species is this generation of a superoxide radical, an oxygen with a free radical electron on it, because remember that electrons want to be in pairs, and when they're not in pairs, they're very angry and they'll interact with anything it can get its hands on. And this radical, this reactive oxygen species, can lead to the generation of hydrogen peroxide, which can lead to a hydroxyl radical. Both hydrogen peroxide and the hydroxyl radical are dangerous to the cell. On some level, they're hydroxyl radical more than hydrogen peroxide, but because hydrogen peroxide can be converted to just water, which is harmless, right? So basically how this works is normally you have a ribosome that is making a nascent protein. That's what this chain is. And there's a molecular chaperone that basically protects this nascent protein. And then it folds, and there's this intermediate of it not being fully folded, but it's with the chaperone still, fold some more, and then you have your native protein. But when there's misfolding, you get a misfolded protein, and this can lead to ubiquitous degradation in the proteasome. This can lead to apoptosis, which is cellular death when the cell decides to kill itself because it's like past any sort of repair. Or it leads to aggregation, which aggregation is when misfolded proteins all clump together. This is very dangerous to the cell because it's like a big blockage in the cell, and things need to transport to and from. And so this can lead to autophagy, which is where the cell basically swallows itself and reuses its materials. Or it can lead to apoptosis. So there's multiple ways that the cell deals with it when there's too many proteins being misfolded, but obviously that's not the goal. The goal of the UPR is to get to this step right here with the native protein. Oh man, this is a horrible image. But how is the UPR activated? So it's activated by oxidative stress. So if you can kind of see this figure, that's horrible. So redox homeostasis, you can look at it like this. So oxidized or aggregated proteins can be, there's not that many of them, so they, when redox homeostasis is normal, and so the proteasome, autophagy, and unfolded response can degrade those aggregated proteins no problem. But when oxidative stress is present, there's more oxidized or aggregated proteins, and on top of that, the proteasome, autophagy, and unfolded protein response is downregulated, which leads to impaired clearance of these aggregated proteins. So I don't know how this is thinking. There it is. So basically, when there's a folded protein and then there's reactive oxygen species that are there, it causes the ROS to oxidize the protein itself. And so this can look like the OHs on the ends of R groups and amino acids getting converted to reactive oxygen species, because basically the reactive oxygen species itself steals the electron from the amino acid, and that's not good. Then this protein becomes very angry and just binds with whatever it can, and that's why it aggregates. And then once something aggregates, the goal is to get it degraded. But the unfolded protein response can take these oxidized proteins, and before aggregation, these molecular chaperones or holdases basically protect the oxidized proteins, so that way other proteins such as foldases and MSR proteins can refold the protein back to its normal state. So there is reversible and irreversible type of modifications that can be done when ROS interacts with a native protein. So reversible ones happen to proteins that have sulfur groups, so that's like cysteine and methionine. It can create tons of thiol type of proteins, but also sulfoxide, sulfonic acid, and glutathionates. And then but these can all go back to just an SH, which is a thiol, which is the native state. But irreversible ones is like making sulfonic acid or carbon or carbonyls, nitrotyrosines, lipid peroxidation, or glycoxidation, which is when you add a sugar to it. And you don't want these irreversible ones to happen, because once those happen, then basically there's no refolding the protein. It's done. It's going to have to be degraded. So we talked about protein oxidation products, but what about DNA ones? Because remember in the beginning I mentioned that the unfolded protein response can be an activator for the DNA damage response. And let me just do something with my computer screens so I can see all you guys. Okay. Okay. So, just like proteins can be oxidized, so can DNA. And so, when DNA is oxidized, it's basically like the hydroxyl group is added. And so all these are not good because basically what happens is another reactive oxygen species can come in, steal this hydrogen, and create a radical oxygen, which is not good. So, what happens when there's this oxidative damage to DNA? So a DSB can occur and SSB can occur. A DSB is a double strand break and SSB is a single strand break. So either both strands of the DNA are broken or just one strand of the DNA is broken. And this causes a lot of genomic instability, which genomic instability is the concept of the genome being unstable because of either like DNA damage or sometimes because of like epigenetic factors. So that's like epigenetics is like when the DNA is modified but the sequence of the DNA is the same. So basically other things that can occur with oxidative damage is you get mismatch pairs or you get helix distortion, and all four of these will activate the DNA damage response in different ways. So the more complex way to heal DNA damage is the canonical DNA damage response, which is this right here, because double strand breaks is the most common kind of DNA damage, and it's also the most dangerous and the hardest to repair. But as you can see, there are many players that play a role in doing this, and versus the other processes have much less proteins that are involved in it. So this just goes to show like how complex this one pathway is. So you might read in papers, they call it the DDR, the DNA damage response, but sometimes the DDR can also be referred to as DNA damage repair. So what's the difference between DNA damage response versus DNA damage repair? Well, the DNA damage response is the cascade pathway that leads to DNA damage repair. And then DNA damage repair is actually fixing the DNA. So the DNA damage response is responsible for recruiting downstream effectors that are going to control the cell cycle, DNA repair, and apoptosis if it's beyond repair. Versus the DNA damage repair itself is going to then recruit other proteins that can actually heal the DNA. And so I mentioned there's different kinds of DNA damage. Well, there's, depending on what kind of DNA damage occurs, there's different ways to fix that DNA damage. There's NHEJ, which is non-homologous end joining, and there's HR, which is homologous recombination. And then there's MMEJ, which is microhomology mediated end joining, which is kind of like the in-between repair of NHEJ and HR. So basically you can think of NHEJ as like there's no template involved in the repair of the DNA. It's just kind of like two ends sticking together. Versus homologous recombination, they use the sister chromatid or the homologous chromosome as a template for repair. And then MMEJ uses a template for repair, but it just uses microhomology regions, which are just found at the flanking sites of the DNA damage spot. So, what are the hallmarks of NHEJ? So, a double strand break, this is actually false. A double strand break does not have to occur via NHEJ, can occur via homologous recombination as well, but it's the more prevalent form of DNA repair of double strand breaks because NHEJ is faster. So NHEJ requires the proteins Ku7080 to protect the broken ends of DNA, and then it recruits DNA PK. So DNA PK is DNA phosphokinase. DNA PK is of kinase, which means it can phosphorylate other proteins, and this causes the recruitment of other repair factors. A hallmark of NHEJ is that it's highly mutagenic because there can be a lot of misfilling in of DNA bases because they're not using a template. It's just like they're stitching it back together, but it's fast, and it's mostly done in the growth phases of the cell cycle, because when there's DNA damage during the synthesis phase, because the chromosomes are already loose and exposed, it's more likely to undergo homologous recombination because there's a template available. Versus in the growth, that template is not as readily available. So, this is just exactly what I was saying. So, this is over time, number of double strand breaks. So, in S phase, HR increases versus NHEJ decreases because NHEJ is more prevalent in G1 phase. But yeah, I don't know what I basically explained what I was trying to explain here. So microhomology and joining is kind of uses some things from NHEJ, so it still uses the so basically what happens, there's a double strand break, but then there's resection, and then these resections basically this region and this region is homologous to like this broken piece of DNA up here, and this region is homologous to like this piece of broken DNA up here, and so they anneal together. It's more mutagenic than NHEJ, and it can occur still even if NHEJ and HR is intact, like those processes aren't interrupted or anything like that. It's kind of just like a backup mechanism for DNA repair. Now, homologous recombination. So the hallmarks of that is that it's error-free, it's a slow process of DNA repair, and it's primarily done in S phase. So how this briefly works is you have the double strand break in one of the chromosomes, and the other chromosome is intact, the homologous chromosome or the sister chromatid. Then resection occurs of the damaged DNA, creating these flanking regions, and these flanking regions then invade the sister chromatid or homologous chromosome and use it as a template for to repair itself, what I'm trying to say. But then there's two different ways that this occurs. It either occurs via DSBR, which is double strand break repair, or SDSA, which is like strand displacement synthesis annealing or something like that. And the more common is the DSBR, because this creates something right here, these X's of the strands invading the homologous chromosome. This is something special called a holiday junction. And this process is just the best in repairing the DNA efficiently and correctly. And the end result ends in a crossover. So remember, from meiosis, crossing over is when the two sister chromatids like swap DNA. So that can also occur during DNA damage repair. But why would homologous recombination occur? Well, it can occur it occurs most times due to problems in the replication fork by either like lesions blocking the way, or neck DNA, or a collapsed fork. So what are the main two DNA damage repair pathways? That's NHEJ and HR. MMEJ, even though I explained that to you today, it's not considered a main repair pathway. It's a minor repair pathway that kind of helps out with both NHEJ and HR by taking some of the workload off of it. But the important things to remember are the hallmarks of it. So NHEJ is error prone, while HR is error free. NHEJ is a quick DNA repair mechanism, while HR is slow and is a long, extensive process. It also depends which way it's going to be repaired, depends on where the cell is in the cell cycle. So G1, G2 phases, growth phases, they're going to opt for more NHEJ, versus homologous recombination, they're going to opt for S phase. Or when it's an S phase, they're going to opt for homologous recombination. NHEJ does not require a template, versus HR requires a template, such as the sister chromatid or homologous chromosome. So yeah, you should now be familiar with these concepts. And now we will talk about this paper a little bit. So what time is it right now? So I asked you guys to come up with 3 questions that you had about this paper, but we don't have too much time. So I'm going to ask you guys to come up with, or you guys can each ask one of your questions. So whoever wants to go first, I'm going to stop presenting this and pull up the paper. Well, I guess first what I'll ask is, what did you guys think? Like, this is your first time having to read a scientific paper. So was it challenging for you guys? Were you able to Google some things and be like, oh, okay, I kind of got it?
I didn't actually get a chance to read this yet.
Okay.
But, it's
That's
not on the
fine.
Google Drive anymore, so where would I find this? It's on the website, right?
Yeah, so we're going to be using the website because my Google Drive, like, basically my storage was full, and I was like, there's so many videos I'm going to have to upload through this course, so, like, not a good option for me to use that, so I just decided to use my website. But yeah, so I'll give you guys the TLDR of the paper. So basically what they're talking about here is in hypoxia conditions, which is low oxygen, hypoxia. Basically, hypoxia is an inducer of replication stress, which elicits the DNA damage response, which then elicits the unfolded protein response, etc. They have this nice figure here kind of showing how the two processes are connected. So in the nucleus, when replication stress occurs and activates the DDR, there are these kinases called ATM and ATR, which then phosphorylate proteins that regulate the cell cycle, such as the check proteins or P53, and this leads to the mobilization of DNA damage repair machinery. So you can see that like DDR here refers to like the DNA damage response versus this refers to DNA damage repair. So those can be tricky. Versus the unfolded protein response, basically you have this cascade of proteins that dimerize, such as IRE1, PERK, or ATF6, and they phosphorylate. This phosphorylation leads to a transcript being phosphorylated and modified and turning into a protein, which then translocates into the nucleus to then upregulate UPR target genes, so that way the ER stress can be solved and the unfolded proteins can be refolded. And so both of these processes are modulating how the DNA is being processed. And so what they're asking is, how is hypoxia activating both of these pathways, and like, are they connected? And they basically just did like a huge literature review on it. See if there's any other cool figures. So basically what they the main takeaways of the paper is that both the UPR and DNA damage response pathways have a significant impact on the cellular response to hypoxia. So if these pathways are damaged at all, then their response to hypoxia is going to be dampened. And there are links between the two pathways that are emerging in studies that use like pharmacological induction of creating redox stress. And they're saying that further research is highly likely to identify mechanistic and functional links between UPR and DDR, which are important in hypoxia. So that's the TLDR on it. You don't have to do anything with this paper, like submission-wise. So if you want to read it and you haven't read it yet, if you're interested in investigating like the DDR and the UPR response, like either one or both, how they're connected, then I suggest reading it. If not, kind of just like move on to the next stuff, because next week there's going to be another paper, so just like read the next week paper. So all you guys have to submit before next lecture, which I'm not meaning tomorrow, because tomorrow is like part 2 of our lecture. It's the R lesson, so it's not going to be me presenting a PowerPoint and a paper. It's going to be me doing basically what I did in that video I uploaded, which is going through the R script with you guys. So by next lecture, so next week, submit the lecture worksheet, and yeah, that's about it. I think the only thing I didn't go over on the lecture worksheet was what's osmotic stress. Osmotic stress is the stress of there being either too much or too little water in the cells. And so, like, too much water can cause the cell to explode, but too much or too little water can cause it to shrink and shrivel up and die. So that's what osmotic stress is. I don't know why I put that in the worksheet. I made this worksheet like a year ago for the last year's course, but yeah. Oh, I realized I presented all that stuff to you about the paper without showing the figures because I didn't have my screen shared. But it's on the video, so if you want to go back and watch this, I was pointing to stuff. But yeah, are there any questions on anything right now, or like how to navigate the website or anything like that, or what you should be doing?
I had a question. I couldn't find the article we have to read on the website.
Let me check. I mean, I may have forgotten it. That's a highly likely probability because I was uploading everything to it. And yeah, you are correct. I did not upload it. So I will upload. So basically, like, I will upload the paper for this week. If you are curious to read it, read it. If not, just let it be. Focus your time more on submitting that worksheet, reading the paper for next week, and then continuing to read the 5 to 6 papers that you choose from my research experiences, because come meeting 5, which is our first meeting that's like not a lecture, we're going to be jumping into like, okay, so what are you guys interested in? So you'll need to be prepared for that meeting, and so I'm giving you these 4 weeks to like slowly work on reading those papers and figuring out some research questions. And from my research experiences document, I give for each one some potential tangential projects that you could do with each one of those topics. So feel free to like steal one of those or offshoot one of those or whatever. But yeah, any other questions?
I've been getting emails like saying that my research proposal is due, but since we like started really late, that's not like actually due until later, right?
Yeah, don't listen to anything that those automated things. Basically, it's your program manager hasn't updated the deadlines based off the syllabus. So
Okay.
go off the syllabus for your deadlines and don't freak out about those automated things.
Okay.
Yeah. I've been in contact with Kabir and they should know what's up, but probably just hasn't gotten to like actually updating the system yet.
Okay.
Any other questions? So we'll meet in a new link I'll make for tomorrow. I have to make all your links. All the GCal events, I just haven't done that yet. And then I'll upload that stuff to the website regarding the papers. Any other questions? Okay. Well, thanks for coming to class today, guys, and listening to me. Yeah, we have 2 more weeks of this, of kind of just like listening to the stuff that I'm teaching you guys to introduce you to these topics, but then I'm just introducing you to these topics so that way it can help better inform you what kind of project you want to do. So don't worry too much about like getting in all this knowledge that I'm throwing at you guys, because this is all like very high level like biochemistry knowledge that we would teach at the doctoral level. We wouldn't even teach this at the undergraduate level. This is not what an undergraduate biochemistry course is like, so this is to give you more real life context as to what biochemistry is like as a field. And yeah. Okay, you guys are free to go.
Thank you.
Thank you.
No problem.